Our next topic of the biochemistry series, it's the Krebs cycle, also known as the citric acid cycle or tricarboxylic acid cycle. And we have discussed plenty in detail about the glycolysis. So let's get started. First of all, I want you to know that this Krebs cycle occurs inside mitochondria. Okay, this occurs inside the mitochondria because what is the reason for that? It is because the enzymes required for this process, okay? The enzymes required for this process are present in the mitochondrial matrix. Let's discuss about some structure of the mitochondria in detail. This is the mitochondria. The outer and the inner membrane are separated by the intermembranous space, and the inner membrane has several foldings known as the cristae, as you can see here. And the process occur inside the mitochondria. Okay, the enzymes required for this Krebs cycle is present inside the mitochondrial matrix, or they can be attached to the inner foldings of the inner mitochondrial membrane. This process is catabolizing the acetyl CoA and releasing a tremendous amount of energy in this process. Okay, as far as glycolysis is concerned, we have invested one glucose molecule and we have yielded two pyruvate, and these two pyruvate are converted into acetyl CoA. We are discussing it in this lecture now. So acetyl CoA is a two carbon compound. As you know that pyruvate was three carbon compound. Okay, but acetyl CoA is a two carbon compound, and with a coenzyme attached known as the coenzyme A. The acetyl part of this compound, the acetyl part of the acetyl CoA, is involved in the Krebs cycle, and with the help of its carbons, with the help of the reactions, we are yielding the energy in the form of NADH and FADH2, and this will be converted into other intermediates of the Krebs cycle. Eventually we get the last intermediate. Why this cycle is known as the cycle? It's known as the Krebs cycle, right? It's the citric acid cycle. Why this reaction, why this process is known as the cycle? It's because that the first and the last intermediate of this cycle, which is acetyl CoA, is the same. In contrast, all other reactions, for example glycolysis or electron transport chain, the first substrate and the last product is not identical, they are different. The first product of the glycolysis is the glucose, the first intermediate, and the last intermediate is the pyruvate. So they are different, but in Krebs cycle, the first intermediate, which is acetyl CoA, and the last product is also acetyl CoA, so that's why it's called as the cycle. So let's talk about something. In this reaction, the energy is generated by the breakdown of the carbon units of the acetyl component of acetyl CoA, right? So carbon components are break down, and this acetyl CoA undergoes decarboxylation. It means that the carbon is removed from this compound, and the carbon is removed, and only thing we get from that removed carbon is the carbon dioxide. Okay, we get carbon dioxide from here. And also this reaction has involvement of dehydrogenation. It means that we are removing hydrogen as well, and hydrogen, where that hydrogen goes, it is used to convert NAD positive to NADH. Here the hydrogen has been removed, and FAD positive to FADH2. Here the hydrogen has been removed. So let's put some basics in your mind that the Krebs cycle involves the reactions of decarboxylation and dehydrogenation. And decarboxylation reaction forms two molecules of carbon dioxide, and the dehydrogenation reaction involves the formation of NADH and FADH2. And these NADH and FADH2 are like reservoirs of energy when they go to the electron transport chain in the presence of oxygen. Okay, we are talking that in the plenty of supply of oxygen, when these reservoirs of energy like NADH and FADH2 goes to the electron transport chain in the presence of oxygen, They are running through that reaction and they are generating a lot of ATP, a lot of energy, and they are converted back to NAD positive and FAD positive. It's known as oxidative phosphorylation because it's involving oxidation reactions and phosphorylation of the components. Also, one thing worth mentioning here that in addition to oxidative phosphorylation, there is also one ATP that is produced at the substrate level phosphorylation in the Krebs cycle. Okay, somewhat here in which the ADP is converted into ATP. There is no involvement of oxygen here. Okay, there is no involvement of electron transport chain. So that's why it is called as the substrate level phosphorylation. So what we have discussed until now, we have discussed that the Krebs cycle involves the decarboxylation of acetyl CoA and dehydrogenation, and decarboxylation will yield molecules of carbon dioxide, and dehydrogenation will yield NADH and FADH2. We'll discuss about the numbers of these compounds as well. And from where the acetyl CoA is obtained, from where the acetyl CoA is, we obtain acetyl CoA. We obtain acetyl CoA either from proteins, because the proteins, carbs, and the lipids, they three have carbon structures in them, right? So glucose have six carbon structure, protein also have amino acid which have carbon, and lipid also have carbon structure. So they are broken down until they are reduced to two carbons. Okay, the two carbon compound is known as the acetyl CoA, and that we use to involve in the Krebs cycle, the tricarboxylic acid cycle or citric acid cycle. And acetyl CoA is the major fuel for the Krebs cycle. Krebs cycle cannot use any other compound or substance in place of acetyl CoA. So that's important. So let's talk about the energy involvement and energy yielding and investing process of this, all of the glycolysis and the tricarboxylic acid cycle. We will discuss in detail about each of the reactions and enzymes involved that, but now this is just an introductory lecture. So let's get started. First of all, in glycolysis, we have in glycolysis, the net production of ATP is 8 molecules of ATP, right? And we have discussed in the previous lecture. So in glycolysis, the net production of ATP is 8 molecules of ATP. We are talking about when one glucose is involved. Okay? When one glucose is involved, the net production of ATP in glycolysis is 8 ATPs. The last reaction in which the phosphoenol pyruvate is converted into two molecules of pyruvate, this is one and this is the second molecule of pyruvate. Okay, and before entering the Krebs cycle, this is tricarboxylic acid cycle. Okay, this is the tricarboxylic acid cycle because we have two molecules of pyruvate, so we will have two cycles of the Krebs cycle. We will have two Krebs cycle from one glucose molecule. Before entering the Krebs cycle, there is also a energy involvement phase. There is an energy involvement phase in which the pyruvate is converted into acetyl CoA by the enzyme known as the pyruvate dehydrogenase, and this is a dehydrogenase enzyme, right? So here the three carbon compound convert into two carbon compounds. So where one carbon goes, it is puffed out as carbon dioxide, right? And this dehydrogenation means that hydrogen is also being removed from here. So where that hydrogen goes, it goes from NAD positive and NADH is formed. So in glycolysis, we have yielded 8 ATPs, right? And before entering the Krebs cycle, we have an energy involvement phase. And here, one carbon dioxide is formed and one NADH is formed from one pyruvate. But from one glucose, we have two pyruvates. So how many NADH are formed before the Krebs cycle? Two NADH are formed, okay? Two NADH are formed before entering into the Krebs cycle. And as you know that one NADH is equal to how many ATPs? NADH is equal to 3 ATPs. One NADH is equal to 3 ATPs. When it goes into the electron transport chain in the presence of oxygen, oxidative phosphorylation is going to occur, and one NADH molecule will going to yield 3 ATP molecules. So from this reaction, how many ATPs we will get? 2 NADH are being released, so 2 multiplied by 3, so there are 6 ATPs up till here before entering the Krebs cycle. So so let's now enter into the Krebs cycle. Because we have 2 pyruvate, we have 2 acetyl CoA. So how many Krebs cycle will going to run? 2 Krebs cycle will going to run. So let's discuss one, and the other will be the identical. One carbon dioxide is removed here. When acetyl CoA enters into the Krebs cycle, it is undergoing the process of decarboxylation. It means that carbon dioxide are being removed, and dehydrogenation, it means hydrogen are being removed. So how many NADH are formed in one Krebs cycle? It's 3 NADH. 3 NADH are formed in one Krebs cycle, and also the energy reservoir compound known as FADH2 is formed. How many FADH2 are formed? It's one FADH2. 3 NADH and 1 FADH2 are formed in one Krebs cycle. So 3 NADH from one and 3 NADH from the other. So how many NADH are produced from 2 Krebs cycle? It's 6. 6 NADH are produced from 2 Krebs cycle, from 2 acetyl CoA, from 2 pyruvate, from 1 glucose molecule, right? We are talking about 1 glucose molecule. 6 NADH is equal to 6 threes are 18. So 18 ATPs are produced from NADH in 2 Krebs cycles. So how many FADH is produced in one cycle? It's 1. So in 2 it will be 2. So 2 FADH2 are produced in 2 cycles of the Krebs, and FADH2 is equal to 2 ATPs. 1 FADH2 is equal to 2 ATPs. Okay, so there are 2, so how many ATPs will be there? There will be 4 ATPs up till here. And why have I written 2 ATPs here? I have told you that 1 ATP is produced by substrate level phosphorylation somewhere here. Okay, somewhere here. So in the Krebs cycle, there is also oxidative phosphorylation. When NADH and FADH2 goes to the electron transport chain, and in the presence of oxygen, the reaction will proceed and they will form the subsequent ATPs. According to, if it is NADH, it will form 3 ATPs per NADH, and if it is FADH2, it will form 2 ATP per FADH2 molecule. But 1 ATP is also produced in the Krebs cycle without the involvement of oxygen, and this is known as the substrate level phosphorylation. It's not called oxidative phosphorylation. Okay, so one is produced here, one is produced here. So how many ATPs are produced by substrate level phosphorylation? It's 2 ATPs. So I have written 2 here. 2 ATPs are produced by substrate level phosphorylation, SLP. So let's count all of them. In glycolysis, we have 8 ATPs in net, 2 NADH before entering the Krebs cycle, 6 NADH from 2 Krebs cycle, and 2 FADH2 from 2 Krebs cycle. So in total, there are 38 ATPs from one glucose molecule and in the abundant supply of oxygen. So it's very important to remember that when mitochondria is functioning, when oxygen is there, when aerobic processes are going on, the anaerobic story is somewhat different, because there is no involvement of electron transport chain, because there is no oxygen, and the pyruvate will be converted to lactate by lactate dehydrogenase, and in glycolysis only 2 ATPs are produced in anaerobic conditions, and there will be no Krebs cycle in case of anaerobic conditions. Only 2 ATPs are produced in glycolysis from one glucose molecule. So in case of abundant supply of oxygen and presence of one glucose molecule, we have 38 ATPs from 2 Krebs cycle. So let's like divide all of them. From glycolysis, there are 8 ATPs. from conversion of pyruvate to acetyl CoA from one like site, if there will be 3 ATPs because one NADH is there, and from 2 there will be 6 of course, and from one tricarboxylic acid cycle there will be only 12 ATPs, but we are talking about 2, so there will be 24. So how there will be 12? Because it's 3, so 3 threes are 9, and 2, 9 to 11, and 1 here from the substrate level phosphorylation, so there will be 12 ATPs from one Krebs cycle. This is the equation for the cellular respiration, in which the C6H12O6, it's glucose. In the presence of oxygen, I am repeating that these all processes occur aerobically. 6 carbon dioxide will be removed and 6 water will be removed, and we are obtaining ATP. And how 6 carbon dioxides are there? Can you mention them? Yes, of course. Conversion of pyruvate to acetyl CoA yielded first carbon dioxide molecule, here the C1. In the process of the Krebs cycle, there will be removal of further 2 carbon dioxide. So 3 from here and 3 from here, there will be total 6 carbon dioxide removal from one glucose molecule. Okay, and carbon dioxide is exhaled as carbon dioxide during expiration phase of respiration, right? And water will be removed from urine, and energy which we wanted, which we are planning to gain, is obtained in the form of ATP. This was just a brief introduction of the tricarboxylic acid cycle, and in the next lecture we'll discuss each of the intermediates and each of the enzymes involved, and other energy expenditures and yielding processes, and we have discussed it now that 38 ATPs are produced from one glucose molecule and two TCA cycles. So this was for today. I will see you in the next video with further detail. Till then, goodbye.